Thanks, Petros, for the introduction. And like everyone else, I'd like to thank the organizers of both this workshop as well as the Lung IPAM program. It's a delight to be here. I'm really privileged to be able to share some of our work with you today. So I'll start by saying that, by acknowledging some of the collaborators in the work that I'm about to tell you about. So the theoretical collaborators are Steve Brunton, Nathan Kutz, and Raj Rao. And I'll be showing you data from a couple of experimental collaborators, Jeff Ogeman and Bill Moody. So before I start, as you probably saw from my slide, I'm a biologist, and I'm going to have this obligation to tell you a little bit about your brain. So there's a lot of talk nowadays about neural networks. So I'm going to tell you a little bit about the neuronal network that is inside each of your heads right now. So if you look at mammalian brains, they have similar structures. They're very different, especially in size, but they have some commonalities as well. Down here is a human brain. It has, to the best of our estimation, about 86 billion neurons. Each neuron has approximately 1,000 to 10,000 connections with other neurons. So if you think about the complexity of that network, it is rather astonishing. So one of the things that I like to do is tell people a little bit about the sizes and shapes of these things that we're talking about. So this is a participation part of this talk. So I'm going to ask everyone to make two fists. Go ahead, you have to do it. This is not optional. Make two fists and put them together. That is approximately the size of your brain. Just look at it for a second. It always surprises me how small it is. Maybe because I have a big head, I don't know. I know some of you have bigger hands than others. Don't take it too seriously. So as you've probably seen these pictures before, the human brain and many of the other brains, but not all of them, are wrinkly. And they're wrinkly because they're approximately a two-dimensional sheet that got stuffed together, jammed inside a skull, and so it got wrinkly because it was jammed together. If you take the neocortex, which is just the surface layer right here, and you shook it and ironed it, it would be approximately the size of a large dinner napkin, like about that big. It is very thin. Petros, do you have a credit card?
Sure.
May I borrow a credit card?
Borrow my credit card.
Okay.
All right. So, anyone ready with numbers 4? I'm just kidding. So a credit card is about 2 millimeters thick, and you can all picture the size of it. This is the surface area, right? This is approximately the size of your primary visual cortex, almost exactly like this. It's about this thick. And if you folded it in half and stuffed it in the back of your brain, that is your primary visual cortex. Thank you, Petros. Yes, you do. Thank you. So that gives you at least some anatomical sense of scale about how big these things are. Now, I get really excited about studying the brain. And here are some of the reasons I get really excited. The brain has multiscale structure in space. So what I'm showing you on the left-hand side are orders of magnitude size scales in log coordinates, down from a micron all the way up to a meter, right? And the remarkable thing about this system is that there is interesting structures happening at every single one of these size scales. So the basic unit of connection between one neuron and another neuron is called a synapse. These are the connections, right? And they have weights. So in a neural network, this would be the W. And it's a lot more complicated than that, but that's kind of what it is. Now, so here is a kind of cartoon of all the proteins that are inside these synapses. Now remember, we have 86 billion neurons, each of which has about 10,000 of these things. There are entire communities of neuroscientists, gigantic meetings, international conferences, where all they study is one synapse. And it's no wonder, because if you look at the complexity of a single synapse, it's like a city. It has highways, it has traffic, it has power plants, and it has garbage plants. So this thing is extraordinarily complicated, and there's many of them, and there's many of them per neuron, and there's lots of neurons, and they all connect to each other in a way that lots of people try to describe. And ultimately, at the order of a meter, it controls your entire body. So, another good factoid, this is neuroscience for cocktail parties. You know, you can next time you go to a cocktail party, you can share with them this fact. The longest cell in your body is a neuron that starts at the bottom segment of your spine. One end of it projects all the way to your big toe. the other end of it projects all the way to the base of your neck. This one single cell is practically as tall as you are. Now, imagine that in a giraffe. Pretty cool, right? But that's not all, because not only does it have multiscale structures in space, the brain also has multiscale dynamics in time. So here, I noticed, I went to a couple of the talks earlier on molecular and chemical kinetics, and you all have the same axes, except it's shifted by some orders of magnitude. I start at milliseconds, going all the way up to years. We don't go femtoseconds in neuroscience, but otherwise it's exactly the same. So at the order of milliseconds, we have the basic unit of communication between neurons, which is an action potential. It's approximately 1 millisecond. And if you look at all of the other things that happen that enable speech, for me to talk to you, I have to coordinate muscle activation in my articulatory muscles on the order of tens of milliseconds. I have to be able to do that in order to talk. And you have short-term memory over the course of minutes. I've been talking for 5 minutes so far, right? So you have to have some memory of what I talked about 5 minutes ago and integrated with what you're hearing now to understand what I'm talking about. So that's on the order of minutes, right? On the longer time scale, there's decision making. There's small decisions like, do I have coffee or decaf today? Always coffee. And there's longer term decisions like, what graduate school do I want to go to? Do I want to take this job or not? There are things that vary with every day, every month, right? And every season. And ultimately, even though every single part of your nervous system, all of those molecules in all of those synapses have a lifetime of approximately a couple of days. Do you remember the picture of the synapses I showed you earlier? That's the basic unit of connections. All of those proteins have a half-life of approximately 3 to 5 days. So every 3 to 5 days, each of those proteins degrades and they're replaced with new ones. So over the course of decades, you are not comprised of the same proteins that you were when you were 5 years old, yet you somehow feel like you're the same person. We don't understand how that happens, but it does. So these are the big questions as I see them in understanding neuroscience. And I think the connection to this conference and this workshop in particular is the observation that somehow over all of these time scales and all of these size scales, all of us manage to learn physics in such a way that we manage to walk around in the world, to make decisions, to go rock climbing, to not fall down the stairs every single time I go down the stairs, and to learn enough of the physics that we do it effortlessly. So it had often been, if you follow any of the work in artificial intelligence and robotics, one thing that often gets said is that it was easier for us to train a computer algorithm to play chess than to build a robotic hand that can pick up the chess piece and put it somewhere else. I think this kind of motor control and the understanding of how do we interact with and manipulate the physical world is very underappreciated because it's actually a more difficult problem. It has proven to be a more difficult problem for us to solve than logical abstract problems. And it's underappreciated because it feels easy to us. We all do it, and we don't even think about doing it, so it feels easy. Things that feel easy don't mean that they're actually easy. And so to give you a big overview of what I do in my lab, and I'm not going to talk about the vast majority of this work, in general, we like thinking about how our brains function in the natural world. How do we interact with each other organisms, and the physical world in such a way that feels effortless? How do animals do what they do best? And we use a variety of different model organisms to do this. So we have a couple of projects on flying insects. Steve talked about this a little bit earlier in the week. I'm not going to talk about that at all. We have some work on rodents and some primates. Here's another primate. I do work on humans, which I'll talk about a tiny bit. This is our daughter Abigail doing what she does best, which is eating spaghetti carbonara. I don't actually study her. I just needed a picture of a human, so there she went. And what ties all of this together is using data-driven dynamic models to understand neural computations, especially underlying naturalistic behavior, how brains do what we do best. Because this is IPAM, I'm not going to talk about most of it. What we're going to focus on are the computational techniques, and especially the models that we're able to build. So I'll use a lot of neuroscience examples as motivation, but as you'll see, and I know you'll appreciate, a lot of these analytic techniques have a lot in common with what's being used in physical systems and chemical systems and all kinds of other systems. So I'll start by showing the type of data that I stare at quite a bit. This is the data. It's a lot of squiggly lines. These squiggly lines happen to come from recordings of a human brain. Each line is one electrode that's in the brain, and I'm stacking them on top of each other just for visualization purposes. There's about approximately 100 of them. And the deflections in the vertical direction are voltage. So this is a view into what this particular person's brain looked like at that particular moment. The time scale of this is a couple of seconds. We have hours and days of this stuff. And what you can see are, even if you just look at a tiny snippet of data like this, you're starting to be able to see that there are patterns in this data. So there's patterns in two different senses. One is there's spatial patterns. I've told you that each of these are electrodes at different brain locations. You'll see that if you look closely at some of these electrodes, they seem to be having up and down deflections at the same time. So different brain areas are correlated. They're kind of doing the same thing at the same time. Furthermore, if you look over time, you'll see that there's periods of it where it almost goes sinusoidal. So for example, over here, there's a couple little ripples, right? And it goes and comes and goes as transient. There's a few more over here. There are a couple of electrodes over here that are kind of going up and down at the same time. Okay?
How many cells is each electrode responding to?
Excellent question. So the question is, how many cells are each electrode responding to? These are larger electrodes. So we're measuring conservatively probably 10,000 neurons at a time. That's approximately what it is. Okay, so I know yesterday was Halloween. I don't know if you saw any gruesome costumes, but in case you didn't get your chance to see anything particularly scary, I'm going to show a picture, and it has blood. So if you get queasy, don't look, okay? So I'm going to show you where this data came from. It comes from a surgery that is done on patients with epilepsy. So this is the clinical need for this procedure. And what the surgeon does is that they cut away your giant piece of your skull, and they open it up just like a hood of a car. They leave a piece of muscle attached, so it actually just opens up. And then they put in this array of electrodes. So each of these electrodes is responsible for one of these traces of data that we're seeing. These electrodes are 2 millimeters-ish in diameter, and this is a 1 centimeter grid. So that gives you a sense of size of how far apart these things are. That make sense? And so since we have all of these patterns in the data, and that we have patterns in space and in time, what we really want to do is reduce that complexity by doing some dimensionality reduction. So how many people here know about DMD? Okay, great. I know you have seen this before, but I'm just going to show it again because I really like this image as a way of building intuition. So let's say I have data of this kind. So here's a movie, and here's a movie that I made, so I know the right answer. And what you're seeing is an 80 pixel by 80 pixel movie. So it's technically 6,400 dimensional data. It's noisy and it varies in time. Now, this data is not 6,400 dimensional. It's much simpler than that. How many things are happening in this movie if you just looked at it with your human eyes? How many things do you see? 2. Good. Could somebody please just tell me in English words, what are the 2 things that are happening? Two frequency oscillations. We all agree with that? Okay, good. Could you describe the 2 spatial patterns? You said a square?
Yeah.
Good. And what else?
These.
And a Gaussian oval, right? Okay, good. Do we all see that? We all agree that there's 2 frequencies of oscillation, and there's a Gaussian oval-y thing, and there's a square thing. So that's the goal. If I give you this movie, you will tell me that there's 2 things that are happening, and you will tell me that it's a Gaussian and it's a square, and here are the oscillations. All right? Now, if you didn't know any better, and I gave you this data, let's say you're teaching an undergraduate class, and I gave you this data, or you give this data to your students, what's the first thing that you would do to get the answer that we are looking for?
Denoise.
You can denoise it. How would you do that? How would you reduce the dimensionality to get that there's 2 things happening? POD, yes, exactly right. Okay, so we do POD. Here's what happens when we do POD. It gets that it's 2 things happening. It does that very well. There's 2 singular values that are not 0. Everything else just goes, it's gone. So that's great. But because the two spatial patterns are spatially overlapping, it'll tell you these are the two modes, and the two modes are the mixtures of the two things that you can see by eye are happening, right? Okay, so let's see if we can do better. What's the second thing you would try after POD and PCA? ICA. Great, let's try ICA. Here's ICA. ICA does better. I gave this talk in front of Terry Sanowski once he shared. So, to its credit, ICA does better. It gets that there's a square, and it gets that there's an oval. However, you'll see that the oval is pretty noisy, and there's still a shadow of a square left over. So you'll know what's coming up, which is that if you apply an algorithm DMD, unlike PCA and ICA, it acknowledges the fact that this is actually a movie instead of a set of snapshots that are independent in time. And it gives you that there's a circle and that there's a square, and it tells you the frequencies of oscillation. Okay? So just to remind you, this comes from fluid dynamics for the study of fluids. And what happens is that you have these data snapshots in time. I'm going to stack them up. I'm going to make two matrices, one starting at snapshot 0, and the second one starting at snapshot 1. And I'm going to write down a linear model of the dynamics where there's an A matrix that tells you how to get from one step to the next. And to do so, there's an SVD step and some linear algebra. And what you get out of it are a set of eigenvalues that tells you about growth and decay and frequencies of oscillation. And you get these spatial modes that tell you about correlations between the measurements. So the nice part is that it pops apart everything that happens in space from everything that happens in time. And that's really nice, and you can write it down that way. Okay? So if you don't remember anything else, the analogy that I usually use is that if PCA and the Fourier transform had a baby, it would be DMD. And it's adorable. So there's a couple of things that I had to learn when I was starting to apply DMD to the kind of data that I showed you before, the brain data that I was showing you before. And after trying it for a little while, I quickly realized that it's because the shape of the data matrix was a bit different than the one that's typically used in fluid dynamics. So in fluid dynamics, you usually end up with these really tall matrices where you have tons and tons of measurements and fewer snapshots in time. But the kind of data that I was working with had the opposite problem. It was the opposite rectangle. It was super long, but I had fewer snapshots. I had fewer measurements at every snapshot. And so I took a hint from Jonathan's 2 exact DMD algorithm and started doing these shift stacks, essentially doing delay coordinates in order to augment the dimensionality of the data set. And that worked pretty well. So if you could hang on to this thought, we'll come back to delay coordinates a little bit later in this talk. The other thing that was really cool was that the DMD spectrum, if you look at these eigenvalues, you can plot them in the complex coordinates. And what I learned from working with neurosurgeons is that I really don't like looking at eigenvalue spectra in complex coordinates. So what they wanted is hertz. They wanted cycles per second. And so that was really nice because we can make a transformation of this representation into frequency, as in 20 cycles per second. And it closely resembled what you would actually get from a Fourier transform. So that was really nice as a representation of the data. So I'm going to show you a couple of examples of how we've used this. The first example is looking at sleep spindle networks in the same kind of data I showed you earlier, this brain data from humans. So this is sleep data. Everyone loves sleep. I love sleep. And this is one of the, as you probably have heard, there's several stages of sleep that you go through, canonical stages of sleep. So you can't go sleep 1, 2, 3, 4, and you go to REM sleep, and you go backwards again. This happens several times in the course of a normal evening's worth of sleep. Each of these stages is characterized by some types of oscillations. And one of the types of oscillations that have been noticed very early on in the human electrographic record are what are called these sleep spindles. And there are these things right here. And they're called spindles because if you look at their envelope, it has the shape of a spindle of the kind that you might use in a loom. It's usually centered around 14 hertz, but there's a range around that as well. So I said that we've known about these sleep spindles for a very long time. If you look at this picture of these recordings, can anyone tell me why this is a really old recording? This is not a modern recording. This is an old recording. How do you know?
The lines are curved.
The lines are curved, yeah? Do you want to explain why? How do you know the lines are curved, therefore must be old?
It's probably on a radial tracker.
Yeah, it's a radio track, right? You put a little pen on a radio tracker, and so it's actually going like this with a paper going around it, right? It's the same data plotted a different way on a computer now, but look quite different. In fact, this was a big thing. I talked to a couple of older, more senior neurologists, and when they went between this type of radio arm recording and computer electronic recording, they actually had to adjust their diagnostics because they were so used to looking at these, they couldn't figure out what to do with the more modern computerized versions, even though it was the same data. That was interesting. So we were looking at these spindles. And what happened with the spindles, I started working with my collaborator, Lise Johnson, who was looking at spindles not only in one electrode, but in all the electrodes across the brain. And what she noticed was that they were not global events. It's not like all parts of your brain were having spindles at the same time. Just looking at the data, she told me it looks like some of the electrodes, some of the brain areas are having spindles some of the time, and some of the time it's these other groups. And so we wanted a way to characterize that. We wanted to be able to visualize and quantify the observation that there were some groups of electrodes having spindles at the same time. So we came up with a windowed DMD method of going about it. So here, again, is the raw data, just a couple of seconds worth of it. I'm only showing you a subset of the channels just for visualization purposes. There's actually a lot more. And what you can see is that there's a group of spindles right here and a separate group of different electrodes that are having spindles right next to it. So we apply DMD in this windowed way, and we're going to pick out every spatial mode for which the corresponding power in the 14 hertz range was more than you expected it would be. By the way, here's a funny factoid. I'm just going to load this up in your brain. People in the physics community fight about this. They've been fighting about this for decades. I don't know what the answer is. There seems to be this decay in brain, which is approximately 1 over f. Approximately, I say approximately. People fight over that exponent. Some people think it's close to 1, some people think it's not 1. There's a lot of theory around exactly what it means that there's a spectrum here that's approximately 1 over f. I don't know what it means, I have no opinion. I'm just telling you that people fight about it. I know that's something that comes up in a lot of other application areas of physics as well when people look at these spectra. So if you have any thoughts around brain spectra, happy to hear it. All I care about is that I did a fit, and I'm going to be agnostic about what this alpha actually is. And I'm going to be looking at modes where it pops significantly above whatever that fit is. And if you do that, and I keep the corresponding DMD modes corresponding to each of those deviations, we'll collect this over the entire record of stage 2 sleep. We do some clustering. We can then look at network stereotypes. We can look at the centroids of those clusters. In other words, these are generally the types of electrodes. These are the networks that are having spindles at the same time. Here's what they look like for one subject and a different subject. So the electrodes are plotted on top of the brain, and their size and color both correspond to the magnitude of the contribution to that network. So you'll see that there's one little cluster of them here, one cluster of them here. They're not necessarily local. Here is one that seems to be more spatially dispersed. It's strongest here and here, but it kind of excludes the area in between. So there's other things that we can do with this, but I just wanted to show you what these networks look like. You can also do this not in electronic records, but on these fMRI records. So these are magnetic resonance imaging machines. And so you get imaging of this kind, which instead of being at 1,000 samples per second, these are more like sampled every couple of 100 milliseconds. So it's much slower, but it's less invasive. It's in fact non-invasive. The other one requires brain surgery. This requires you to lie inside a big magnet. So you can do the same analysis where we take windows of this data. And here, because we have non-invasive recordings, you can actually get a large number of subjects, much larger numbers of subjects. But the idea is basically the same. You run window DMD, and we cluster them. And after you cluster them, you can get these stereotypes, these networks that seem to be coherent and oscillating at approximately the same coherent frequencies at the same time. We can do this at a group level by putting all of the subjects together, which works great if you're studying neurotypical brains. We can also do it at the subject level, where you do it for individual people. And this is useful because when you're studying things like neurodegenerative disease or anything where it's a pathological case, you want a set of networks that are personalized to that particular brain, because you can't necessarily average it and agglomerate it with other brains because you don't know if they're similar or not. The other thing that you can do with the subject level clustering is you can get a subject level diagram of the transitions between the various networks. And we've shown that that works pretty well. So I've already told you everything that's on the slide. I just wanted to show it because it's kind of pretty. So it's a bunch of DMD modes that we've clustered. And you can see that there's all of these. There's a lot of structure in these modes that we agglomerated over some 120 patients, 100 patient subjects, I should say. And here are the things that come out. OK, so now we're going to go back to DMD. And as you all know, if you think about it, it's just a regression model in the dynamics. It's pretty simple, right? You take every data point is x at time k versus x at time k plus 1. And we're just writing a regression model. And so we were talking about this with a couple of students. And one of them started thinking about, well, if you look at it from the perspective of doing regression, I mean, people have been doing statisticians, especially, been doing regression for a very long time, right? There's a lot of knowledge we've learned about how to do regressions. And one of the things that puzzled us was the following. Oftentimes, we don't want to be looking at the dynamics around 0. We want to be looking at dynamics around fixed points or equilibria. And what that really looks like is you really want your dynamic model to have an affine term. Because usually there's an affine term in your regression model. If you have the data, but you don't include an affine term in your model, you get totally the wrong answer. And that was puzzling for us, because in the generic DMD model that we talked about, there is no affine model. It's closer to this picture. This is just a cartoon. And what we've shown is that if you wanted to incorporate an affine term, it's equivalent to doing the regression on center data. So this is the work of Seth Hirsch, a graduate student that I co-advised with Nathan Kutz and Cameron Decker-Harris, who is a postdoc. And what they showed is that centering the data improves the DMD. So this is a slightly subtle result. So I'll show you. I won't go into it in the detail. Everything's on the archive if you care to read about it. What we can show is that if you have a linear system with no affine term, doing DMD with and without the centering works just fine. They both work. However, if you assume there is an affine term, then DMD without centering only works some of the time. And the details about exactly what the criteria are for making that work are in the paper. So some of you may or may not remember that this is not the first time people have looked at centering in DMD. So we compared our results to an earlier paper by Kevin Chen. And what we were able to show is that their formulation of the DMD with centering came from the companion matrix formulation of DMD. And if you use the exact DMD formulation that's based on the SVD, the results are different. So if you look at their version of the results, you do get this convergence where the eigenvalue spectrum converges onto the unit circle. But that does not happen if you use the exact DMD SVD formulation. So that's one of the differences between what we did and what they did. So that works as long as your model is pretty linear. You either have a linear system or a linear affine system. We also applied it to a couple of nonlinear systems for which we know the model is wrong. And what you can see here is for Lorenz, and this is only one lobe of the Lorenz, the true data is plotted in blue. And if you do DMD without centering and DMD with centering, both of them are wrong. But if you do centering, it works better. So this is more obvious in the bottom here, where the blue is the right answer. And I'm trying to fit the blue nonlinear model with a linear DMD model. Without centering, it's this orange trace, which basically just crashes to 0 pretty quickly, like it should. And if you do centering, it's this green curve here, which does better at following the nonlinear dynamics. Is there a question?
Yeah, so you mentioned you did window DMD. Do you move the center then to the window?
Thank you for asking that clarification question. In this example, we're not doing the windowed version, but I'll come back to the window version later. So this is only per window. Yeah. There's also a generalization as a result that Seth figured out, which is if you think about centering, it's kind of equivalent to subtracting the zero frequency of oscillation. Once we figure out how to do that, we figure we can also subtract arbitrary frequencies, as long as you know exactly what they are. So one of the problems that I think about when we're doing electrical recordings is, in the US at least, there is always 60 hertz noise, because that's the noise of the room. You can often actually tell by people's data where they did the recordings based on the line noise that's in their recordings. So my recordings have 60 hertz noise, because they're from Seattle. And so since we know it's 60 hertz and we know it's exactly 60 hertz, we can subtract out the 60 hertz noise from the data. And so here's the data with 60 hertz noise. It's these large deflections here that are totally not real. They're not in the brain. And since we know exactly what they are, we can subtract them out. That's kind of an interesting application. So I'm going to go back to the windowing, because I think that's a really interesting point. One of the ways of dealing with the fact that DMD is a linear method and we want to fit a nonlinear world is to acknowledge, well, if you look at a small enough window, it's kind of linear. Right? So if you want to look at really long data, you can just do a window DMD technique, like we've been doing for the brain data. So this is the work of Cameron Decker-Harris. And what Cam realized is that if you think about window to DMD, right, we're fitting these vectorized autoregressive models in every window. And if you fit them over many, many, many windows, and you do it independently, one of the issues is that your modes don't really line up. They're totally in different bases every single time you move the window over. So he formulated it in the following way. If you chunk up your data and you cut them into windows of data, there's an A matrix for every single window. And if you stack it up over all the windows, there's actually a tensor, right? So instead of solving for each of those frontal slices of the tensor independently, we're just going to solve for the entire tensor. And the way he formulated it is the following. We're going to assume that this tensor is low rank. And if you assume it's low rank, then we're able to solve this problem. And I'll tell you a little bit about how we did that. So the model is the same. It's a vectorized autoregressive model in every single window. And we're going to assume that this script A tensor thing, it looks like the following, where it's low rank. It's a rank R. Right? So there's R of these multiplications. So here's how it works. You have windows of data. And in each window K, you have the same formulation as before. There's an X matrix and a Y matrix, which is your data. So we're going to be solving a least squares optimization problem, again, exactly the same as regular DMD, where this error is what we're trying to minimize. And if you wanted to get your A matrix back, A sub K, each A sub K, you can get it back from the solution. Okay, so we can go back and forth. There's an equivalence here. And you can see that this is starting to look a little bit like the SVD, where there's a left vector and a right vector, and that's the formulation of the problem that's going to come out. Now, this alone is not sufficient to solve the problem. So there's a couple of tricks that we had to use. One of them is that it's pretty massively under constraint. And so the coefficients kept blowing up. What we had to do was, in addition to having this least square error term, this is the actual cost function we ended up using. There's also a Tikhonov regularizer, which makes it so that all of the numbers in these U1, U2, and U3 tensors tend to be as small as possible. So you have to encourage them to be small, otherwise they just blow up. The other thing is really interesting, I thought, because this is an opportunity. We are fitting them over all of these windows of data. And if you have a sufficiently small window size and you believe your temporal dynamics between windows has a certain distribution, you can actually add another regularizer on only the temporal tensors. These are the three tensors that are in the window direction. And depending on what you think your data ought to look like, you can choose from a couple of different types of regularizers to encourage your answer to be of that form. For example, if you think that your data should vary smoothly, you can use a spline penalty on U3. If you think your data might be switching between a couple of distinct states, for example, you can use a TV regularizer. And there's other tricks that could be played along those lines. I'll also point out that this algorithm has two parameters. This one here and this one here. There used to be 4, but we got it down to 2. And so there's definitely a bit of tuning here. You still have to be in the right regime in terms of both of these regularizers where you won't get the right answer. So that's the formulation. We've applied it to a bunch of just cool problems that are totally unrelated. It works on worm behavior. So these are C. elegans worms that are about 1 millimeter long. They crawl around, and they have a couple of canonical trajectories of how they're squiggling around, and so we can find that in the data. This works on sea surface temperature data, where you can actually find a mode here. This is the El Nino mode that pops out by itself. Again, this is an entirely unsupervised analysis. All we've decided was we picked those 2 parameters, and we picked R. We picked the rank of the system, and we waited for the whole thing to converge. And so this particular temporal mode has so these are this is a U3 tensor, the one tensor that's in the temporal direction. And it goes up and down in a way that strongly resembles the El Nino cycle. You can also do on a mocap data of just monkeys turning around, and you can segment different types of behavior out of it. I'll show you one more example because it's kind of cool. So this is a totally different data set now, where instead of doing electrical recordings, we're doing calcium imaging. This is optical recording of brain activity. The setup is really interesting. So this is work of Dennis Tabuena, who is a graduate student like collaborator Bill Moody, and they're right downstairs from my lab. And this is a developing mouse pup. And it is so young that it's blind and it's deaf and it's hairless. And its skull is so thin that you can image its brain activity right through the bone. You don't even have to take the bone out. So you just open up the scalp, you put it under the microscope. It has what's called GCAMP, which is a calcium indicator. It's a very bright calcium indicator protein. So we can image calcium activity right through the skull. At the same time, because it's so little, it's basically falling asleep spontaneously. It's waking up for a couple of seconds, falling asleep, and then waking up and falling asleep. So we can tell when it's asleep and when it's awake by putting another electrode on its neck, in its neck muscles. So when it's tensing up its neck muscles, we know it's awake, and when it's relaxed, we know it's asleep. And that's kind of all it does. It's all entirely spontaneous activity. Now, what I love about this, just like with the large-scale cortical recordings that we were looking at earlier in the humans, is that even though we're not getting single neuron resolution, you're getting a sense of the general spatial temporal dynamics that happens over the entire brain, which is something that, as neuroscientists, we don't spend a lot of time thinking about it because it's a limitation of the technology. So I'm really excited about these technologies because I think it allows us to answer qualitatively different types of questions than we were able to ask before. So here's what the data looks like. And to remind you, this is the surface of the brain. Give you a little bit more orientation. This is the midline, so this is the midline of the brain, and we're looking at just the left half, the entire left half of the brain. So this is the front, that's the back, and this is the midline right here. These dark lines are blood vessels. So they're shadows in the imaging, you can't do anything about it. You need blood in the brain. And what you're seeing is a false color movie of the calcium activity. Which parts of the brain are really active at the same time? Down here is the neck EMG. And in this super cool 80s font, Dennis has added on top a W or an S to indicate whether or not the mouse was asleep or awake. So awake, asleep. W is awake, and S is asleep. So you'll see when it's asleep or awake. I'll show you the video. So here it is awake. There's a little bit of activity. That's a sleep wave. There's these giant waves that take over the entire cortex. There it is again. Even though this is in a mouse, we actually have some pretty good evidence that this does happen in humans. There's been some EEG recordings of human infants, and these recordings are consistent with what's happening in a developing human brain as well. So even though you don't remember it, I certainly don't remember it, I think there was a time in our past when there were these giant storms that go over your entire brain. And part of our study is to figure out, what does that mean? How can we understand it? So that's a larger study, but I did want to show you one thing, which is the work of Nate Linden. Nate's an undergrad in my lab. He's been very talented. And Nate's applying to grad school right now. He's really interested in studying computational sciences for his PhD. So I just wanted to highlight his work here because he's terrific. And what Nate's done, one of the things he's done, is used CAM's tensor decomposition on the calcium imaging data. So here you're seeing the 3 of the modes that pop out of the decomposition. The first mode is usually just the average, same thing as happens in SVD. And we're plotting here modes 3, 4, and 5. And so this is the trajectory of that data projected onto these modes. And what you can see is that most of the time nothing is happening, it stays near the origin. And occasionally, there is a large deflection that corresponds to one of these waves. And there's one. That's what it looks like. Okay? So we've now talked about DMD, and there's lots and lots of people, including many of you in this audience, who have tried to develop extensions of the technique to acknowledge the fact that we are intrinsically interested in nonlinear dynamics, but we are stuck with a lot of the frameworks that are easiest to deal with analytically when they're linear. So we're going to keep talking about this idea. So I'm going to stand on my soapbox for a second and make the following statement. One of the reasons that your brains work and the reasons that deep neural nets work is because when you expand the dimensionality of the inputs, it makes the representation more linear, or at least it can, right? So this is popularly known as the kernel trick in machine learning. And the dynamical systems version of that, one of them at least, I would say, is the Koopman operator. It's this idea that if you increase the dimensionality of your representation of your dynamics, things that used to be tangled up and nonlinear might become more linear. So why is this a good thing? This is a good thing because it makes many things easier, like classification, in the case of the kernel trick in SVMs. It makes it easier to model dynamics because we can use tools like DMD, and it might make it easy to do control as well. And the reason I think this might be related to my core research in neuroscience is because I actually suspect that a lot of the control tasks that animals solve when you're solving a complicated sensory motor task, like hiking along the riverbed, right? You're an inverted pendulum stepping on round rocks that may or may not be slippery, and you're trying not to fall over. This is a massively nonlinear task. And in fact, the interactions of every single time your foot touches the surface as opposed to when you pick it up, I mean, that transition has been very, very difficult to model for roboticists. Yet, again, you do it effortlessly. You don't even think about it. You can go hiking and it's not a problem, right? So I think part of the reasons that deep neural networks have been so successful and the reason that networks and neurons have been so successful is because we increase the dimensionality of the representation of the world in such a way that not only classification and models are easier, control is easier. So in the Koopman framework, it's related to DMD in the following way. So you have some complex system, which is unknown. We have no idea what it is. We don't even know how to represent it. But what you do know are measurements. You can measure data. DMD takes those observables directly and hits it with a linear regression model. The Koopman perspective is a little more nuanced, in which case you say that there are these nonlinear observables, and you don't necessarily know what they are. But if you knew what they were, then you can build a dynamic model on top of it that is linear, and that's great. So we're going to use this idea to try to understand a canonically difficult nonlinear chaotic system that everyone knows and loves. I just wanted to show a cool video that Steve made, where this grid of red dots are being integrated forward in time. And you can see that they start out together, but very quickly they diverge and spread out through the butterfly attractor. I will also note that I am actually on the floor of the biology building that's full of entomologists, and butterflies have 4 wings. So for the record, this is not a butterfly attractor because it only has 2 wings. Okay, just wanted to say that. Okay, so we want to understand this. And we want, if you were to model it using more of a DMD or Cindy type framework, you would have the following. You would collect your data, because it's 3-dimensional, so you have X, Y, Z. And you might have derivatives, again, in x, y, z. And if you were doing a DMD model, then this would be a 3 by 3 tiny little matrix that you would be building to model the system. And we know this is not going to work, right? We all know this. Let's not even try, because it's not going to work. We know this is massively nonlinear, even though we did try earlier with Seth's stuff. Furthermore, you may not even know that the system should be measured in x, y, and z. Maybe you only have x. Maybe you have some random combination of x, y, and z because you didn't really know how to look at the system. This is much more reasonable, right? When you're looking at a new system, you may not even know how to represent it. What are the natural coordinate systems to represent it? So let's say that you only measured x, but it could well be a random combination of x, y, and z, let's say. So we measure x. And what we're going to do next is build this time delay coordinates. And we're going to say that we're going to put everything by building this Hankel matrix. So instead of just having x as a time series, we're also going to stack it up with copies of itself delayed by 1 dt. And you can build this thing to be pretty tall if you wanted to, right? Next, we're going to take the SVD, which is our best friend. And what that gets you is a representation of the same dynamics in time delay coordinates. And this idea has been around for a very long time, going back to the 80s with the tokens embedding. So that part's not new. Steve wanted to talk about this earlier this week, and he didn't quite get to it, so I get to talk about it. What you do get to do is then build a linear regression model in the coordinates. So in the coordinates of these u vectors that come out of the SVD of the Hankel matrix. And if you do that, something kind of remarkable happens. Even though this is a massively nonlinear, chaotic dynamical system, it turns out that you can build a darn good linear regression model for most of it. And it's only the very last term that's a bad fit. And it's interesting in the following way, because the linear dynamics actually work really well. And the intermittent forcing, which is the bad fit part of the model, it's not on all the time. This is not like I built a terrible model, I'm going to call everything else I couldn't account for in the residual, even though it kind of is that. But these forcing terms have the interesting statistic in that they're not Gaussian. They're 0 most of the time, and they only turn on some of the time. So this works for the Lorentz. It also works for a variety of other synthetic as well as real data examples that we applied it to. And what's interesting is that if you look really closely at the very last term in the Havoc model, what you see is that the forcing term is active in a way that predicts the switching, is a lobe switching. And what you see is here that if you color the portions of the trajectory for which the intermittent forcing term is very large, it precedes the switching by a little bit, which we thought was pretty cool. Again, you can do that for all of the examples. So here I've added a column to highlight the nonlinear regions of these trajectories in each of these systems. You can see it kind of matches what your intuition would be. So for example, in this one here, where we're looking at a model of the switching of the Earth's magnetic fields, you see that there's a region here, which is one orientation, and the region here, which is the other orientation. And the red highlights all of the transitions between these two states. The part I skipped over is this linear model. So there's this A matrix that is a linear dynamic model of most of what's happening here, barring the intermittent forcing term. And if you look at it, this is not a generic linear model. It has a very special structure. It's a skew-symmetric matrix. And so for a long time, we wrote this paper, we're kind of going like, I don't know what's going on here. Why? What's happening here? So there's a couple of subtleties here, which is that if you look at this matrix, it doesn't show up on the projector very well. But if you look at the numbers in these off-diagonal blocks, you can kind of see it because I'm standing really close to the projector. These numbers out here are not exactly 0. So that was a mystery as well. It's like, okay, so It clearly has a structure, but is this some kind of numerical thing? What's going on here? Why is it not exactly skew-symmetric? And what does it mean that it's skew-symmetric? So something that we've been really excited about is, again, the work of the very creative Seth Hirsch. Seth figured out that the SVD of time delay coordinates and building these linear regression models in this way is related to geometric mechanics and curvature in Furness-Area coordinates. In other words, we figured out an interpretation of why this matrix is skew-symmetric. We now have a way of making it exactly skew-symmetric instead of just approximately skew-symmetric. And we now have an interpretation of what all of these coefficients might mean. So he's in the process of writing that up, and hopefully you'll be able to see that soon. OK, so if you don't remember anything else from my talk, here's what I think I do. I think in my day job, I work on data-driven dynamics and models of all kinds of high-dimensional time series data, especially those from brain and behavior. Kind of a mouthful, I know. A couple of years ago, our son came home from kindergarten. And you guys have you've either done this before or you've had your kids give you these before, where on Mother's Day, they write a little book for you, and it's all really sweet, right? He comes home, and here's the book that he gave me. He says, my mom and I like to do math, and my mom really likes brains. I think he meant to say brains instead of brands. But you know, this is a pretty good drawing of a brain, right? It's not bad. It could also be a bran flake, so I'm not really sure. Maybe I really like fiber. But what I really do is I have the privilege of working with this group of tremendously talented students and postdocs. I haven't had the opportunity to talk about what most of them do today. I only highlighted work mostly of Seth and Cam today. But they're great. And what's really great about them is that they come from all kinds of different backgrounds that are not like my own. And I get to talk to them, and I get to learn all kinds of stuff that I did not get to learn in the course of my own training. So that's been really terrific. So thanks very much for your attention.